Hi, everyone. Hi, Richard. You can hear me okay there?
Yes, I can.
Okay, great. Well, welcome to our webinar today. So I want to thank everyone for joining us for our live webinar, which is going to be presented by Professor Richard Gross, entitled Lipidomics and Phospholipases. My name is Valerie O'Donnell. I'm based at Cardiff University in the UK, and the webinar is being recorded and it'll be available on our YouTube channel and our website following this talk. We got about 45 minutes to 50 minutes for the talk today, with the remaining time up to an hour to answer your questions. And if you got a question during the talk, please put it in the chat or the Q&A. And what we'll do at the end is we'll ask you to unmute yourself, and then you can ask the question yourself to Richard. So we'll take as many questions as we can in that time. And the webinar today is sponsored by Cayman, and we are grateful for their ongoing support. So I'd like today to introduce our speaker. So I'm delighted to introduce Professor Richard Gross, who is really one of the pioneers of lipidomics, mass spectrometry, and its biomedical applications. Richard received his AB degree from Columbia University and his MD from New York University and a PhD from Washington University. He had the privilege of being mentored by Dr. Gilbert Stork in synthetic organic chemistry at Columbia, Burton Sobel in cardiology, and David Kipnis in diabetes and metabolism. So really spanning the whole gamut of chemistry right up to clinical investigation, which is an absolutely fantastic training for the world of lipidomics and its biomedical applications. So he trained, after his cardiology training, he matriculated into the chemistry department at WashU and was mentored in magnetic resonance, and was a Searle Scholar and the recipient of an established investigator award from the American Heart Association. During the last 40 years, Dr. Gross's laboratory is known particularly for his work on lipidomics, intracellular phospholipase, mitochondrial function, and the roles of membrane molecular dynamics and physical properties in modulating transmembrane protein functions. Richard's been a pioneer in the development and application of lipidomics methods, I think it has to be said. Indeed, he was one of the primary leads in the emerging field of shotgun lipidomics back in the mid-2000s, working with Shanlin Han. I know he's going to be talking about this today in his talk, which will be really nice to hear. More recently, he's been working on a field of research that's very familiar to me and my lab, and that's relating to enzymatically oxidized phospholipids. Indeed, his recent work on platelets, showing how enzymes, long-known enzymes, make oxygenated lysophospholipids through novel mechanisms, is opening up significant new avenues in our understanding of how lysophospholipids are metabolized and how they may signal in innate immunity. I'm looking forward very much to hearing more about this work today. Richard has authored over 200 peer-reviewed publications, and as a clinical investigator, I should say he was the PI on a 20-year program project grant on biochemical mechanisms of diabetic cardiomyopathy. So it's been a massive focus of translating basic science and chemical characterization of lipids into clinical practice, which is really, really wonderful program of work. So currently, Dr. Gross is director of the Division of Bioorganic Chemistry and Molecular Pharmacology and a professor of medicine, chemistry, and developmental biology at Washington University School of Medicine and the Washington University Danforth campus. So we're delighted to welcome you here today, Richard, and looking forward very much to hearing your talk. So I'll hand over to you now.
Thank you very much, Valerie, for the kind invitation and the overly kind introduction. It's true that I spent many years with different professors, and it was a real blessing to get so many different perspectives on what now has become a very important and rapidly developing field in membrane dynamics, membrane changes, and their roles in a whole host of membrane delimited events. So I first became an assistant professor and a cardiologist, a freshly minted cardiologist in 1982. And like all younger people, I had very lofty goals. I wanted to cure sudden death. I wanted to cure heart attacks, prevent heart failure. And I was immediately attracted to the membrane, especially the sarcolemmal membrane, which is the site of numerous ion channels that are part and parcel of the ion currents carried by ion channels. There were receptors that affected heart failure and transporters, which determined the membrane polarization, the degree of membrane polarization. And this is not news to anybody, but there's a diverse repertoire of chemical species present in the membranes, which can be released and demonstrate their cryptic activities. The membrane is also an important a special place for self-organizing chemical assemblies. And it's a storage depot for lipid second messengers that we've all come to love. So I worked for about 2 years, right? Homogenizing hearts, trying to do column chromatography and find out what makes the sarcolemma so special, and what phospholipases are involved in changing either the molecular dynamics, chemical composition, surface charge of the sarcolemmal membrane. And I will readily admit, in the first 18 months, I got virtually nowhere. And my friend, Jeff Staffitz, who now is at Harvard, pointed out to me what turned out to be a very inconvenient truth. Although we were looking for things that caused arrhythmias and altered sarcolemmal characteristics, this electron micrograph on the right demonstrates unambiguously that about 80% of heart lipids, 80 to 85% of heart lipids are mitochondrial lipids, and that these mitochondrial lipids completely overwhelm lipid analysis of the sarcolemmal membrane, which is only about 1 or 2% of membranes. So my first efforts were directed to developing a purification scheme for these sarcolemmal phospholipids, with the understanding that the things that we were doing the phospholipase assays with, that were purchased from New England Nuclear, may not be the same physical constituents or chemical constituents that are present in the sarcolemmal membrane. So we decided we needed to first purify the sarcolemma and then identify its chemical composition as best we could. And this led to a couple of very interesting surprises for us, which I think set the path for future studies. So we developed a method to purify sarcolemma from canine ventricular myocardium. It was enriched about 100-fold, 80 to 100-fold in specific activity of sodium potassium ATPase. And here you can see a fast atom bombardment mass spectra of choline glycerophospholipids on the left and ethanolamine glycerophospholipids on the right. And I'd like to point out several groups of peaks here. Okay, what was surprising to us was that there were a number of major peaks, okay, and that wasn't really the way we were thinking, although perhaps we should have been. And the ethanolamine glycerophospholipids were a complete surprise. Shown here, highlighted in yellow, are the ethanolamine glycerophospholipids. These, the moieties at MZ 724, 750, 752 are plasmalogen molecular species. For those of you who are unfamiliar with the subclasses, we have the diester phospholipids, the plasmalogen phospholipids, and alkyl ether phospholipids. They're discriminated by the type of bond, right, at the SN1 position. Now, what was remarkable, in addition to finding that the majority of the sarcolemmal membrane was comprised of plasmalogens, was the fact that all these plasmalogens, right, had arachidonic acid at the SN2 position. So in many cell types, if not in most cell types that have anything to do with electrical activity, plasmalogens represent from a very large portion to a majority of phospholipids present in a cell in the ethanolamine glycerophospholipid class. So this was very interesting. So it was obvious that having this cis double bond would change the geometry, right, of the molecule. And so I went back to some of my training and tried to understand what changes plasmalogen phospholipids made to the dynamics and structure of the membrane. And this was, largely due to one of my better ideas, which was the fact that I recognized that H white star, shown right over here, and H blue circle, okay, were always going to be 2.4 angstroms apart from each other, because if they're in plane, there are no rotamers to consider, and they're like the perfect internal standard for looking at through space interactions through dipolar coupling. So the experiment that was done, and Zhang Han, my first graduate student, did this experiment, where we irradiated H white star, okay, and as expected, the relative nuclear overhauser intensity increased very dramatically. Also, it was shown that irradiation of H white star communicated with H blue circle, H green circle, and H yellow circle, showing that they were within 4 angstroms, right, of the radiated proton. The trace on the bottom shows that it is due to spin diffusion, because there's not an exponential increase upon the initial kinetics of dipolar coupling during irradiation. So now we had, I think, pretty definitive proof, okay, that the structure shown in plasmalogens on the right, okay, and we have H white star communicating and within 3 angstroms or so of the proximal portion of the SN2 chain. This stood in direct contrast to the structure that was believed to be that of diacyl phospholipids based on hydrated crystals. Now, hydrated crystals are not the same as liposomes or circular membranes, but it was good enough to make the comparison. And then we subsequently went on in a lot of work that I will not show today to show that plasmalogens and diacyl phospholipids have different motional characteristics, rotational correlation times, different relaxation times. And we were particularly interested in moving this project forward by looking for phospholipases, which could distinguish the difference between plasmalogens and diacyl phospholipids, especially in light of the fact that the plasmalogens was a major carrier of arachidonic acid moieties in many cell types. And you can see here that the phospholipase is going to cleave it into arachidonic acid, lysoplasminylcholine, or lysophosphatidylcholine. And we were interested in different phospholipases because if there was truly backside attack of this carbonyl at the SN2 position, it differed substantially from the type of structure and binding to the phospholipase that would be present in plasmalogens. So we went ahead and we purified fractions that were enriched in hard phospholipases, now using plasminylcholine as substrate. The plasminylcholine was synthesized by us through dicyclohexylcarbamide condensation with the deuterated or spin-labeled lipid of choice, and we got very intriguing results showing that although the specific activity was very low, which I'll explain to you the reasons for in a couple of minutes, there was about a 3-fold increase, okay, in plasmalogen hydrolysis compared to phosphatidylcholine hydrolysis in small unilamellar vesicles. So now having shown an activity, and this is a calcium-independent activity, a neutral active calcium-independent activity, which was big news at the time, is not such big news now. But it led us down the path of wanting to purify this enzyme and find out how it works, what influences it might have on the physiology of a heart cell. At the same time, concomitantly, Lori Loeb, working in our lab, was able to purify a sheep platelet phospholipase. And this is a a tracing of after 3 steps of purification, and it's a mono-Q column. So as you can see, most of the activity, okay, is H open circle, and that activity corresponds to plasmalogen molecular species. Okay, H filled circle, okay, is the amount of activity you get with diacyl phospholipids. Okay, and the integrity of the assay, the integrity of the substrates were proven not only by independent GCMS and NMR methods, but also by the fact that where there are a lot of phospholipases hanging out at around 150 millimolar concentration, all 3 labels, so the diacyl label, the plasmalogen label, and the alkyl ether label, all were hydrolyzed to very similar extents by the enzymes eluting right at about 150 millimolar. But for some reason that was unknown to us, okay, the CPLA2 activity, the calcium-dependent activity, okay, eluted very, very late in the column. This was also the first demonstration of a phospholipase that could be activated by nanomolar amounts of calcium. One of the things that I learned in medical school was the resting concentration of calcium inside a cell is around 50 nanomolar, and it can go up to 200 to 400 nanomolar, very different from that described for other phospholipases, especially the low molecular weight phospholipases, the secretory phospholipases, the venom phospholipases, which all required 100 micromolar or more to determine or assess their full activity. So this slide emphasizes a calcium-independent phospholipase, a phospholipase found in platelets, which demonstrates very marked substrate specificity for the nature of the bond at the SN1 position, and the fact that it was remarkably sensitive to calcium ion. So with moving further, and this will be what I'm going to talk about mostly today, will be what we call IPLA2 beta and IPLA2 gamma. And these are part of a patatin phospholipase family, which is the PNPLA2 designation. And these moieties were connected by the fact that they had a very homologous active site, okay, and also contained various decorations, especially in IPLA2 beta and IPLA2 gamma, which thrilled us with excitement because we thought these would lead to new findings that would be practical to understand biologic processes and disease in humans. The other family of phospholipases are the cytosolic or the CPLA2 phospholipases, where they have a calcium or a lipid binding domain, usually diglyceride, sometimes PS, and they all had the same homology to the active site. Now, one of the differences between the CPLA2 and IPLA2 family is that the CPLA2 family has a lid over the active site, which can be overcome by different concentrations of salt, as we published years ago. CPLA2 gamma doesn't require calcium, yet it plays a prominent role in the hearts. And CPLA2 epsilon and zeta are enzymes which I will be coming back to later in the talk. So these are the main phospholipases. These guys are triglyceride lipases. And then this is PLA7 is a lysophospholipase that was first found by Rudy Zechner. And PLA5 is a lipase that's present in the vasculature, which influences the outcome of human hypertension. So as we call this fondly in the lab, these are the families. Now, why was the specific activity of the heart cytosol so low? One reason was that when you dialyze cytosol, so first of all, you homogenize the heart, okay? And then you dialyze it. And we did this for almost 6 months and we got nowhere. And finally, we recognized that you need to have EGTA, glycerol, and ATP in the dialysis buffer in order to retain activity. So we got a 100-fold increase approximately from that. And not only is the enzyme stabilized by ATP, initial rate kinetic analysis shows that there's about a 5-fold increase in reaction velocity through comparisons of vehicle alone compared to ATP. And it wasn't due to a phosphorylation event on the enzyme, since ATP gamma S was without effect. The other quite remarkable thing that we found was that calmodulin, in the presence, that calcium-activated calmodulin, can bind to the enzyme and completely inhibit it. So in the presence of calcium, this enzyme becomes inhibited quite differently, right, than the IPLA, right, than the CPLA2 family, which requires calcium, and that acyl-CoA can reverse the calmodulin inhibition. So you can activate it by calcium, and the activation can be removed. You can inhibit it by calcium, and the inhibition can be removed by acyl-CoA. So I'm going to talk a little bit now about a 25-year-old enigma in IPLA2 beta, which has been called a signaling enzyme by our group and a housekeeping enzyme by others, just trying to change the nature of the SN2 substituent. So when Stan Hazen purified this enzyme to homogeneity, it was a 40K protein. However, later, using sequence from Simon Jones' patent, we were able to clone, right, the gene, right? And in the gene, we were able to express large quantities of IPLA2 beta. So what's the difference in these steps? So the difference is that hearts have tremendous amounts of calpain. And when you do the tissue homogenization, there's all sorts of calcium release from internal calcium pools, mitochondria particularly, okay? And the 80K holoprotein is cleaved by calpain at a consensus cleavage site that is present in all forms throughout evolution of this enzyme. And it goes from an 80K protein to a 40K protein. And let's look at the activities, okay? So this is SAPC, steroidal PC. And you can see it has the typical 3 to 4-fold, it has a 3 to 4-fold bias toward hydrolysis of arachidonic acid-containing phospholipids compared to POPC. But the big winner, and much to our delight and pleasure, was the plasmalogen SAPC analog, which now is getting into, specific activities after calpain treatment of about 12 to 25, I guess, on our best shot. And that's as hot as we could ever make this enzyme. And it comes from calpain cleavage. Calpain cleavage is initiated after myocardial ischemia for a sufficient period of time for the mitochondria to become dysfunctional and release calcium. So this answered, at least for us, a lot of questions about what was going on, about why we had a 40K purification product, because we purified the active site because we were following activity, right? And then the gene results, right, showing that it was an 85K protein, were also true, but it had very low specific activity and did not demonstrate much of a preference. By the way, you can see that POPC, despite what a lot of people say, has equal amounts of activity as an SN1 lipase and an SN2 lipase. So we were very excited about these enzymes and the modulatory sites within them, the differential molecular dynamics they manifested. So the first thing we wanted to do was get a crystal structure on the enzyme. And much to our delight and surprise, this crystallized on the very first attempt. Now, these are beautiful looking crystals. However, when I showed these to my radiology friends, they reminded me that the more beautiful a crystal looks, the less well it will diffract. And that, in fact, was the case with these crystals. They diffracted very, very poorly. And after multiple attempts, we failed. So we decided, Sergei Korolev and I decided that we should send IPLA2 beta into outer space on the Discovery, what an appropriate name, on the Discovery Space Shuttle, where it could be crystallized in a gravity-free environment. Unfortunately, this did not work, but through continued pursuit by Konstantin Mali and Sergei Korolev, we were able to get the crystal structure. Sergei was able to get the crystal structure through selenomethionine substitutions. And let me point out several salient areas. There are 8.5 anchoring domains. This one, the last anchoring domain toward the N-terminus, being the putative binding site for ATP. Okay, there was a sequence that corresponded to a nucleotide binding site. The geometry and the crystal structure doesn't suggest that, but having this sequence basically the same throughout evolution makes me think that there may be some nucleotide effects on this. And finally, the catalytic cysteine was present at 465, and the catalytic dyad was comprised of serine 465 and aspartate 598. Also, we were able to identify a 1914 hydrophobic motif at the C-terminus, which was responsible for calcium binding, as well as the canonical IQ motif, which is present in about 80% of comodulin binding proteins. So now that we've looked at the anatomy of the primary structure, let's see how it looks in space. So the protein actually crystallized as a dimer. And with its dimeric structure, you can see that the active site serines are in close spatial proximity. And when it's cut at the calpain cleavage site around here, okay, it now then exposes two interwoven catalytic sites, which facilitate trans-acylation. And this was very intriguing to us. So getting rid of one of the anchorin proteins increased the activity very dramatically, and was up to the numbers that we had anticipated from our turnover analyses. Now, this series of modulators was very exciting for yet another reason. In 1986, Putney described capacitive calcium entry, store-operated capacitive calcium entry. And basically what this means is that after an agonist binds to its receptor, it will release IP3. Okay, and then IP3, right, is a calcium channel and will deplete the endoplasmic reticulum pool of calcium. When calcium is depleted, IPLA2 becomes activated, and activated IPLA2 produces lipid second messengers. So it could produce, hundreds, maybe even several hundred lipid second messengers, including lysolipids, eicosanoids, delcosanoids. I mean, the list is long but distinguished. But one important thing about store-operated calcium's entry is that with CPLA2 alpha, the enzyme that most people have studied most intensely, in the cells that we have used, the CPLA2 cannot be activated by intracellular calcium alone. The calcium has to come from the outside. So if you do your incubations in EGTA and there's no calcium in the external side of the membrane, then CPLA2 will not will not get activated. However, it plays a big role in activation when calcium is present in the extracellular or space or when the calcium is present in the meteor and goes through these store-operated calcium channels. Now, it's been debated for a long time what these lipid second messengers might be. And recent progress in the field has shown that STIM1 is an ER component which interacts with ORA1, ORA2, ORA3, which Trevor Shuttleworth called the arachidonic acid release channel. So it turns out that the channel that is present for signaling, right, the extracellular calcium entry, is related, right, both in terms of arachidonic acid metabolism and lipid second messenger generation, as well as to amplify existing signaling pathways. As a cardiologist, this was an experiment that I found really delightful. So we made a transgenic animal with expressing IPLA2 beta. The mouse, for reasons unknown to me, has very little IPLA2 beta in its heart. This is very different than humans, than large mammals. So we decided to anthropomorphize the mouse, right, by putting in a transgene for IPLA2 beta. And so what we're doing here is we're using a hanging heart preparation and monitoring the premature ventricular contraction frequency. So PVCs are a pretty good surrogate for damage and electrical instability in the heart cell. And as you can see, with the typical time course of arrhythmias in humans after a heart attack, from 5 to 20 minutes, this is the period, right, where most of the arrhythmias occur and the patients die. Whereas, right, with the regular mouse heart, there are hardly any PVCs. Even more remarkable was the fact that you can completely ablate the PVCs by treatment with bromoenolactone, which is a mechanism-based inhibitor. Now, I want to stress that bromoenolactone can inhibit many compounds, but the fact that IPLA2 beta by itself can initiate arrhythmias that could be blocked by beta, we thought was significant. Not only that, but treatment with Bell also completely inhibited the total episodes of ventricular tachycardia, which leads to sudden death in humans. So having worked on the CPLA story, we realized the importance of identifying the proper substrates and synthesizing the proper substrates and analyzing them in an uncomplicated and high throughput fashion. So we developed something, a set of technologies which we had put under the rubric of shotgun lipidomics. Basically, shotgun lipidomics is comprised of 4 components: multiplexed extractions, direct infusion, and intrasource separation, multidimensional mass spectrometry by looking at precursor ion scans or neutral loss scans or an array in multiple dimensions, and deconvolving them through bioinformatics. So the basic flow is you just get an extract of a biologic sample. You could directly determine anionic lipids in it. For example, PI is some PG. If you add just a touch of lithium hydroxide, you could do a negative ion mode spectra again, and you could bring out all the phosphatidylethanolamines. And of course, in the positive ion mode, the moieties containing a positive charge, a permanent positive charge, such as acyl carnitine and PC, as well as adducted triglycerides can be easily visualized. This whole thing, if you were, like running a race course, this whole thing could be done in 20 minutes, which was, like a far stretch from doing two-dimensional TOC, scraping, right, the spots, and then doing molybdenum polyphosphate assays. The way that the best advantage from the shotgun lipidomics approach was to use precursor ion scans for different head groups, and then neutral loss scans for triglyceride analysis. And you could see that in both cases, right, this is MS1, right, which will go to the detector, and then you could look at MS1, and then you could scan for different biologic fatty acids. So the first thing that we did with this was we demonstrated that shotgun lipidomics identifies the chemical mechanisms of diabetic heart disease. So let's look at this spectra in some detail. On the left is a control mouse, okay, and on the right is a diabetic mouse. These are all normalized by ratiometric comparisons to 17:17:1, which does not exist in nature. The first thing that should be obvious is that the control mouse, right, had a very low abundance of M-STAT products in this region, the triglyceride region, whereas the diabetic mouse had a reasonable amount of ions, right, in the triglyceride section. So what we did was to try and deconvolve, like what fatty acids were present in these triglycerides. We scanned with 16.0, 18.1, 18.2, and here's the big winner. Okay, so you could see that the arachidonic acid is released in diabetic myocardium. The arachidonic acid is present in the diabetic mouse heart, and by extension, you could say that these larger amounts of triglycerides have a longer turnover time, and now that the polyunsaturated, there's targets for free radical mediated oxidation, either enzyme mediated or iron +2 initiated. The second piece of evidence, and this was pretty dramatic, we thought, and this was done with my first graduate student, Sang-Ling Han, as has many of the slides before, and I can't give him enough credit or express my thanks for, working with him for 20 years. We had a good time together. So these are the internal standards, okay, for the control mouse and the diabetic mouse. And you could see that the poor diabetic mice, right, don't have any or hardly any tetra-18.2 cardiolipin. Okay, they also have polyunsaturated fatty acids, which are larger than that present in the non-diabetic mouse. So diabetes gets rid of cardiolipin, and cardiolipin is known to stabilize the respiratory complex of complexes 1, 3, and 4. So this seemed to be a likely generator of mitochondrial dysfunction in diabetic myocardium. So next we asked, is it streptozotocin-induced diabetes, or is it diabetes itself from having a high-fat diet or being obese? And here the results were even more dramatic, is that the OB mouse, which does not have any leptin, basically has virtually no tetra-18.2 cardiolipin and is severely depleted. The fourth portion of evidence that we derived from the shotgun lipidomics technology that was developed was that acyl carnitines, okay, this is the control mouse. This is the diabetic mouse made diabetic by streptozotocin. Okay, so the long chain acyl carnitines went up by, I don't know, say, fivefold. And you could reverse these changes within 2 weeks just by feeding the animal or injecting the animal subcutaneously with insulin. So we were pretty sure that the central portions of diabetic cardiomyopathy were due to mitochondrial dysfunction and lipid second messenger generation. So after having struggled for many years trying to find the smallest membrane compartment in the heart, somehow I came to my senses and said, well, if, like 50% or 40% of mitochondria by volume are present in a heart, why don't we go look and see what's going on in the mitochondria? And this was greatly facilitated by sequence in the pre-genome period that was spit out by NIH on computers, and we found a sequence that was highly homologous to IPLA2 beta, right, and had a mitochondrial localization sequence on it, as well as a peroxisomal sequence on it. There are a handful of proteins that have both peroxisomal and mitochondrial localization sequences, probably originating out of their evolutionary past. So, when we had the sequence of IPLA2 beta, we developed a gene targeting strategy, and this was done by Harold Sims and Bev Gibson and Dave Mancuso, where we took the wild type allele. The active site is E5. So we took the wild type allele, okay, and then basically used traditional methods to flox the enzyme, leaving us with the null allele, right? E4, no E5, no active site, no activity. Okay, and to convince you of the integrity of the specific knockout, I show you here, these are northern analyses, wild type, okay, knockout in kidney, wild type knockout in heart, wild type knockout in brain. Okay, and this was a northern, this is a western, okay, heart, wild type. These residual bands here are due to the presence of vascular cells, which are not induced, which precombinase is not present in because it targets only cardiac myocytes with this MHC promoter. But I think this is pretty convincing too. The wild type heart, the knockout heart, and the skeletal muscle looks the same in wild type and knockout. All right, the kidney looks the same, the brain looks the same. So having this animal in hand, that led to a whole new array of questions that we can answer. The first, and probably one of the most important, was that when people have heart attacks, their degree of recovery and their likelihood of having a normal life depends almost in entirety on the amount of infarct that is present. That is, if you lose a quarter of your heart, you'll still probably be okay. If you lose 50% of your myocardial mass, then you're probably going to be bedbound or, like have difficulty tying your shoelaces. And 40% of myocardium is not, of viable myocardium is not compatible with life. So in a series of experiments with in vivo myocardial ischemia with the control, the wild type, and the knockout mouse, there are three features one uses for infarct sizing. Okay, the first is the area at risk, okay, shown by the red lines here. Okay, so this is all the area that is fed by the left anterior descending artery, which is demonstrated by an injection of dye into the anterior descending artery. The infarct area is highlighted by the yellow lines, okay, and the left ventricle is highlighted by the black lines. And this is the right ventricle over here, which accounts for about 5 to 10% of myocardium in the heart that is not in failure. So we did many of these animals because we thought this was like really important. And the area at risk to the left ventricle was approximately the same. The infarct area to the left ventricle decreased by almost 60%. And doing the gold standard metric, the infarct area to the area at risk, we showed about a 60% decrease in infarct size. So this is, like quite wonderful to be able to reduce an infarct even by 10% is something that is, like heart is worked on with great importance and has great importance. But to be able to reduce an infarct area by 50% is going to make a lot of people return to their normal lives. Okay. So we learned how to cure mouse heart, right? Mouse heart ischemia. What about human heart failure? So getting human hearts, as you might imagine, is a difficult thing to do, right? We were fortunate enough to be located just minutes away from the Midwest Transplantation Facility, where we could get control hearts. These hearts are not perfectly healthy, but they're also not hearts that have been subjected to great degrees of ischemia, and they are not failing. So we looked using charge switch derivatization methods that were developed by Shenping Liu in our lab. And I have to give credit to John Bollinger and Mike Gelb, who made the amino methylpyridine that was used, amino methylphenylpyridine that was used. This turns out to be a great charge switch derivatization moiety because it could undergo charge remote fragmentation. So each of these bars here represent like no calcium, right? This is control with EGTA, no calcium, control with calcium, right? Control with EGTA, and then control with calcium. So there's actually, like a lot, right, of these moieties, of the heats that are formed in human myocardium, which came somewhat as a surprise to us. And what was even a bigger and perhaps nicer surprise was that failing human myocardium could not make 14-15 EATs, which have been implicated previously in increasing ischemic damage. Furthermore, we took some of these hearts and made mitochondria out of them and treated them with bacaline, which will prevent, right, all or almost all enzyme-mediated oxidation. And then we could add back either 12 heat or 15 heat, and we saw that myocardial swelling increased. So it looks like these heats and likely other bioactive metabolites have a great deal of significance in opening up what's called the mitochondrial permeability transition pore. Now, I mentioned before that we were surprised by the amount of CPLA2 present in human myocardium because the mouse hardly had any. And so what Sung-Ho Moon did was he purified this membrane-bound protein. It's bound to the mitochondria. So I thought I learned my lesson in years before is never to go after a membrane-bound protein because it will make your life miserable. But we and to Sung-Ho Moon's credit, persisted. The first step was extraction of material by alkaline exposure, generating membrane sheaths from which the enzyme could be extracted. We then used a DEAE column, a mono-S column, and what you're seeing on the left is the last mono-Q column. So this is 5 steps later, right? And we see bands that we identified by proteomics, okay? And then we did chemoproteomics with a fluoride derivative, right? So that like some nerve gases, because all nucleophiles will react with carbonyl fluorides. And after chemoproteomics, we concentrated it with biotin precipitation. And you can see here, okay, 3 bands which precisely correlate with activity. shown here is bands 25, 26, and 27, right, which precisely match. But even more importantly, when we did proteomics on the purified product, we had two peptides, two large peptides, which had precise Y and B ion patterns indicative of this enzyme not being CPLA2 alpha, but rather CPLA2 zeta. And this also was a shock to us. It turns out that arachidonate has bidirectional effects in heart health, that you need some of it to make, how can I say, to help the heart run physiologically. But obviously, when you get too much of this enzyme or this enzyme becomes inactive, that could be a real problem. So the sort of cartoon that's depicting in the last 5 minutes or so of the talk was that in control myocardium, you have mostly CPLA2 zeta, okay? And then when you go to ischemia or failing myocardium, you have mostly IPLA2 gamma, okay? And IPLA2 gamma makes heats. Heats lead to MPTP opening, and that leads to heart failure progression. Just as importantly, okay, the opening of the MPTP completely depolarizes mitochondria and lets out a whole variety of metabolites that can block the potassium channel. And when this potassium channel is closed, it sets the stage for action potential dispersion, which means that an electrical impulse cannot travel in the same 360-degree direction that it did before, okay? It's blocked. It's blocked because the channels are rendered inactive when the MPTP is opened. So this leads to arrhythmogenesis because they're now short circuits, and you can have a cyclical ventricular tachycardia. And the other thing that was very important from these studies was that mitochondria from human heart synthesize a whole variety of cytokines. I'd like to take just a moment to remind everyone what forward genomics is. So forward genomics is the loss of enzyme function and resultant alterations in phenotype. So that's what's commonly done, is you find something interesting, you knock it out, and then you see what the phenotype alterations are. Reverse genomics is like what Francis Collins did, right? Where he tracked a phenotype, the chloride channel, to a specific gene or group of genes, which were responsible for cystic fibrosis. But this analogy doesn't only extend here. I'd like to introduce the concept of forward lipidomics and reverse lipidomics. Forward lipidomics is loss or gain of a function of an enzyme that's relevant to lipid metabolism and subsequent alterations in the lipidome. Reverse lipidomics is phenotype alterations in the identification of the loss or gain of function of an enzyme or group of enzymes. So the next few slides will be about forward lipidomics. So much to our surprise, when we take 2-arachidonyl LPC, right, which is generated by the SN1 specificity of IPLA2 gamma, okay, you can make bunches of different adducts of lysolipids covalently linked to eicosanoids, and we call these eicosanoid lysolipids. But also, you can make heats. There are probably about 30 to 40 products that we've observed. This is just a small list here. We don't know the complete chemical structures, only that the list of things that are being produced by cyclooxygenases is far greater than I think anyone appreciated. This is particularly important because cyclooxygenase inhibitors are being given routinely to patients with inflammatory disorders. And if you look at a docking experiment, you say, well, how can arachidonic acid, get in there? So this is arachidonic acid attached to phosphocholene. And over here you have the 12 and the 15 carbons that become hydroxylated. So what this does, what this docking experiment showed, was you can have the same Gibbs free energy, or even slightly improved Gibbs free energy, when the arachidonic acid can find its way to the active site, and then you get increasing selectivity and diversity when you can change the polar head groups. Here it's choline, you can do ethanolamine, phosphoinositides, and I'd just like to remind everyone that this is due to SN1 lipase activity. And in fact, all the enzymes that we've ever looked at have some degree of SN1 lipase activity. IPLA2 gamma has the largest. CPLA2 alpha has about 10%, that's the lowest. But everything else lies in between. And there's a high content of 2AA lysolipids during cellular stimulation. So what happens, right, to these lipids? So on the left, okay, this is the control. Okay, now you're adding 12 LOX, 2AA, LPC. Okay, that makes the hydroperoxide LPC, and then reduction with triphenylphosphine, okay, generates 12 LPC. Exactly the same story is true for LPE. Okay, so now you're generating lysolipids, which have eicosanoid moieties. It's esterified to the SN2 hydroxyl. And this is basically the same thing, but showing that it's also possible with heats. Okay, this is the control in yellow. We add 15 LOX, it makes a lot of 15 LOX LPC, and then this could be reduced, hydroperoxide, and this could be reduced by triphenylphosphine to 15. Now, how important is this compared to the amount of non-esterified eicosanoids that we see in a cell? So I think this is something that we've worked on now for probably a year, generating enough knockouts, causing diabetes, and then perfusing them using a Langendorf heart preparation where you basically perfuse retrograde through the aorta. So these are the moieties that we know and love historically, right? 12 heat and 15 heat. And they basically, go up, I don't know, maybe 10%, right, compared to what's happening here. But in hearts that are rendered diabetic, okay, there is a very large increase, probably a 10-fold increase in these eicosanoid lysolipids, particularly 12 heat, right, and to a lesser degree, 15 heat. These alterations, most importantly, can be markedly attenuated by cardiac myocyte-specific IPLA2 knockout. So this is a pathway, basically, for previously undiscovered pathways for lipid signaling, generated from 2AA-LPC or 2AA-LPE. You can see that the normal variety of enzymes works on them. And after they're made into heat LPCs, they can be reesterified, as Gallian Lang has shown in our lab. There are also substrates for LPLC and LPLD. And now you have a whole bunch of interconversion, which has made the problem of metabolic flux in compartments that much more complicated. For probably 35 years, I've been looking for a plasmalogenase. In every new experiment or incubation that we do, we try and check to see if the plasmalogen bond is cleaved. And in fact, we made the remarkable discovery that if you take cytochrome C, right, and so this is background hydrogen peroxide, does nothing to LPC, and with cytochrome C, okay, also hardly anything happens, but cytochrome C plus hydrogen peroxide, okay, is a very robust enzymatic mediator of plasmalogen hydrolysis. Using specific derivatization techniques, we showed that the co-product of this reaction was alpha hydroxyaldehydes. Same thing is true basically for LPE. So this was an enzyme that we've been looking for, I don't know, for my 35 years. And Foilgen, who discovered plasmalogens in 1917, also had his eyes on finding out how plasmalogens are metabolized. So what is the mechanism of plasmalogen hydrolysis? Well, plasmalogens have a state or a resonance structure which is stable where there are two unbonded electrons. Okay. If you take this moiety, right, with a stable resonance state, let iron oxidize it to get a carbon-based radical, oxygen will readily add to that, forming a hydroperoxide, which then can decompose to alpha hydroxyaldehyde and the lysolipids with great alacrity. So this is what the standard pathway was, and we all know and love this pathway for decades now. And this is what we believe is the new pathway and the most relevant pathway, where membrane plasmalogens can be cleaved by cyto C plasmalogenase or SN1 specific lipases, both generate 2AA-LPX, right? Any head groups possible. Okay, and then in the presence of oxidative enzymes, generate an icosanoid lysolipid, which then can be regenerated into saturated phospholipids by either an SN1 acyl transferase or in the conventional Land cycle an SN2 acyl exchange. So we have oxidized phospholipids, which have been known to be DAMPs and death compounds and basically toxic moieties. So here is an even bigger picture, right, of how complex this diagram gets. Okay, if we start with arachidonyl phospholipids, we could generate arachidonic acid, and this is what we all know and love. Arachidonyl phospholipids, as many have shown, can be directly, for phospholipids with 15 LOX, can be directly oxidized. 12 LOX is much more selective. The novel pathways that I'd like people to at least think about are the generation of 2AA lysophospholipids, their oxidation, okay, and then their roles in generating either icosanoids or serving as specific ligands for certain receptors with a higher affinity and different molecular dynamics. Okay, so along that score, let's look at the functional roles of icosanoid lysolipids in inflammation. And here we're taking human monocyte cells grown in culture. These are THP1 cells. And we're adding, you know, like a whole bunch of different oxidized lipids. And as you can see, the only ones that gave a significant signal, okay, were those containing two 12S heats, esterified to LPE or LPC, and also the 12S heat, okay? So anything with an S heat, okay, in this assay will generate TNF and basically initiate inflammation. Now, the question is was this a transcriptional? The question was this a transcriptional event or a translational event? And as you can see here, there's absolutely no change in the amount of message after treating with the bioactive lysophospholipids. So this must be a translational event, and translational events in response to acute inflammation are not uncommon, right? Because you're just stuck somewhere on the mRNA and all you have to do is finish it, right? It could be done much more rapidly and it's certainly shorter lasting. And the critical point in identifying these molecules was looking at the stereospecificity. stereospecificity of TNF alpha. The stereospecificity of TNF alpha shows that in the blue and the orange, okay, the 12S heat, or even the 12S heat LPC, right, are the most highly potent, okay? Having an EC50 of around, say, like 10 nanomolar, 10 to 50 nanomolar. In contrast, the R heat the R heat is not effective, okay? So what this graph is showing you is that there's about a 10-fold preference in terms of concentration preference for the lysophospholipids. This is also buttressed and supported by the observation that if you add 12S hydroperoxide, 12S heat phospholipids, you generate the 25 kDa membrane form of TNF, as well as the released 18K soluble form of TNF, which is used to amplify inflammation. In another aspect, okay, you can see that addition of 12S HP to 12S heat LPC or LPE, okay, generates phosphorylated NF kappa B, okay, and phosphorylated IK kappa B, here and here. And this is a graph showing the amount of NF kappa B per tubulin. And you can see that 10 nanomolar 12S heat doesn't work quite as well as 12S heat LPC. So in summary, okay, I think that we've sort of expanded our perspective of how important the regiospecificity of hydrolysis is, how important cytochrome C is in hydrolyzing plasmalogens and contributing to the 2AA lyso pool. Okay, and this is basically what we've been working on for the last three years or so. Okay, and it leads to TNF alpha either by the new pathway or by the conventionally envisioned pathway. To the best of my knowledge, 12S heats have not been known to activate TNF alpha by themselves, but they certainly do, and it's stereospecific, as I showed you in the graph before. And with that, I'd like to thank all the members of our division who have worked tirelessly to make these observations possible and to increase our understanding of heart disease, arrhythmias, and inflammation. Thank you very much for your attention.
Thank you, Richard. That was excellent. I have loads of questions, but I'm going to only ask one first because I can see there's many others in the chat. And it's really about the two competing pathways in platelets. That I think there's a way to address this actually, because, first of all, you measure a heat P or a heat PC and you don't really know, is it coming through the acylate, which is the substrate for LOX. And we've got an old paper where we used 18O water during platelet activation, because if you replace the buffer water with 18O, then you get heats that are +2 mass units. So
we're doing these experiments as we speak.
Okay. Well, I need to go back and look at the data because it's been published about 10 years
when you use
ago.
the water, right, if it's coming off and becoming free arachidonic acid. So the thing I worry about, maybe you can help me with this, is when you have an H2O18 water, can that exchange and rapidly, like with water, does the exchange rate of the 18O, right, and just water, does that rapidly exchange to make it 16O?
I don't know. We made up the buffer with 18O water. So the only bit of water that should be there is what's in the cells originally. We would hope that by incubating the cells in this buffer, it should get exchanged enough that the dominant one is going to be 18O. So we didn't, we got rid of most of the water as much as we could and replaced it with heavy water. So hopefully it was out competing.
I agree with you about the appropriate strategy to answer this question. And we have the isotopes and have talked
We should
meaning
look at the
COVID sort
paper
of got in
because,
the way,
yeah, I
if
mean,
you know, if you know what I mean.
well, the data's in the JBC paper, and from what I remember, we were hoping to see 50-50 labeled and unlabeled phospholipid, because you'd expect to lose 50% of the label.
Exactly.
I don't think we got 50%. I think we got less. So if we got less, then it suggests both pathways are probably going on. But the data's there, and I'll get it out and I'll email it afterwards and we can have a look, because if it was less than 50-50, whatever is missing of the 50% is probably the pathway you were talking about. And I think that it's very likely both are going on. It'll all depend on where the LOX is, what substrate it sees, the relative concentrations of the two substrates, and their competition with each other. So again, I don't know, and maybe it prefers free arachidonate, but if there's not much of that around, it'll then start metabolizing the lyso forms. So, it could be a time course dependent, like early on you get more from one pathway versus another. But anyway, there's so much to do, and that was really interesting.
There's so much to think about: compartmentation, location, specific activities, time course,
all of it. Yeah.
Right. It's very complicated. There's one additional piece of information in that the IPLA2 gamma knockout platelets, right, they make less oxygenated metabolites. When you knock out IPLA2 gamma in platelets, this is a germline knockout.
Yeah, but that could still implicate either pathway, couldn't it?
I'm sorry?
That could still implicate either pathway, could it?
That experiment, that experiment could be indicative of either pathway, but at least in theory, shows a prominent role for IPLA2 gamma in icosanoid signaling in platelets.
Yeah, and of course, the other thing is that you could get a sterification of the free one in, and then you could get another PLA2 hydrolysis to form the lyso with the heat on it. Yeah, we got no idea. These things are probably continually cycling around between all these forms.
Well, my guess is we've talked about the land cycle for years, and this is like the repeat of it on the SN1 position.
That's very nice. Yeah. Okay, so I better let some other people have the floor. So first question is, what have I done there? Shamshad Cockcroft. So I just need to allow you to talk. Shamshad, you should be able to speak now. Unmute yourself.
I wasn't very clear whether your IPLA2 beta was localized, because you seemed to imply that it was in the lumen of the ER when you're talking about stim ori. Do we know where it localizes?
Are you going to repeat that, Valerie?
Where does the IPLA2 beta localize in the cells?
In cardiac cells. Where is the IPLA2
cardiac cells. So
beta?
you're talking about IPLA2 beta or IPLA gamma or any beta?
No, I'm talking about beta.
Okay, so beta has been localized in many different compartments. Alexander Ma localized it to the outer membrane of mitochondria. We find it mostly in cytosol. It's also present on the Golgi. It's everywhere we looked, we find it. But the majority of it is cytosolic.
Okay. In one of your diagrams, you put it in the lumen of the ER when you were talking of stem and ori. And I got that was very confusing because it's not in the lumen, right? It's in the cytosol.
No, so there is IPLA2 beta in the lumen of the ER. I can't necessarily say it's in the lumen. I can say that it purifies with ER markers.
Okay, thank you. So Ali Hajaya, you've got two questions. Can you just ask one?
Oh, it's difficult to just choose one.
Well, I'll cut you off if you try and ask two.
Yeah. Does COX-2 catalyze the oxygenation of lyso-PS with arachidonic acid?
So lyso-PS24, is that a substrate for COX-2?
Right. So that's something else that that's high on the list. All the head groups are potential candidates for this. And I guess I didn't say it during my presentation, but sort of the fantasy is by having the head groups present, right, that gives an increased degree of discrimination, right, of where the enzyme will sit and what it will sit next to, right, by like having ionic interactions of a phosphorylcholine head group or the ethanolamine, like forming it's just like an endless quandary, right? Of there's 30 well-known polar head groups and I don't know, hundreds of different oxidative reactions. This seems to be a mega problem.
So we have a helicopter landing outside. We're at a hospital site here, so try and ignore that noise. Okay, Roberto Angelini, go ahead.
Hello, Prof. Quick question. I don't want to sound critical, but can I ask you what is the lowest concentration of hydrogen peroxide that you tested in vitro, which is required for the plasmalogenous function of cytochrome C, and if this is actually measurable in living matter?
Okay, so the lowest concentration of hydrogen peroxide was 20 micromolar. But in the paper, if you would read the paper, you see what we did was we oxidized cardiolipin, right, which is a known mitochondrial lipid, right, and very, very small concentrations of oxidized cardiolipin can activate cytochrome C.
Okay. You know, it's still the micromolar concentration of hydrogen peroxide. It's something that, you know, it will be required also for oxidizing cardiolipin by cytochrome C. And
right. So we prepared the cytochrome C by overnight oxidation, and then separated different oxidized components just to look to see if there was a difference. And most of the components work the same.
Okay, thanks.
Okay,
we just put in iron. To oxidize cardiolipin, you just put in iron, leave it on your bench overnight, and then do an HPLC, and you get a bunch of different Cl oxidation products.
Okay, so one last question from Rodrigo de Faria.
Hello, thanks for presentations, very good. I have a question about the other ether phospholipids, sometimes called plasmonil. It's saturated at the SN1 position.
Yes.
In the beginning, you showed the plasmalogen is very reactive with phospholipase. And how about this saturated ether phospholipid? This also have the same reactivity or is lower?
Yeah, so the graph that I showed you of the CPLA2 alpha purification, that was done with 1 plasminyl 2 oleoyl glycerol phosphocholin.
Mm-hmm.
Right, so it's not dependent on the arachidonate being there, although almost all plasmalogens have arachidonic acid attached to them, except, right, the peripheral nervous system, which has di-18-1 plasmalogens. And the reason for that is probably because they make a better electrical insulator, okay, than the diester phospholipids do. And we've actually shown that looking at passive ion currents, right, in just vesicles. We load up the vesicles, we dilute them, and the plasmalogens, you know, like form a very much tighter surface, okay, than the diacyl phospholipids, and have a lower rate of, and plasmalogens have a lower rate of loss of ions inside.
And what about the ethers without that double bond in the plasmalogen? I think that's...
So we, since I showed that paper, we've sort of ignored the alkyl portion of this. There's lots of work to be done. As you know, platelet activating factor is really mysterious, and I'm sure that alkyl phospholipids will find their place in space and time in due course.
Yeah, that will be interesting. Okay, that's great. Thank you, Richard. So I think on that note, we've this has been one of our longest webinars because there's been so many good questions and so much really inspiring work from Richard today. So I've really enjoyed it, and I know the rest of the audience will have too. So thank you again very much, Richard, for joining us today.
It was my pleasure, and thank you for the invitation.
Yeah, and I'll email you all the rest of my comments and questions afterwards because I have many more.
Oh, please do.
Yeah, I will, right. Yeah, and everyone else can too. So thank you, Richard.
Yes, please. If anyone has any issues with, you know, things that I said or statements that I made, or confirm me to things that were done previously, please tell me. You know, I'm really interested if there have been other perspectives of this, or I'm not aware of some papers which were important.
Thank you. Goodbye now.
Bye-bye. Thank you.